Okay, I assume you can hear me okay. Let me try to advance this. All right. So mass spectrometry-based large-scale proteomics using multidimensional separation, is becoming a mainstream workflow for a lot of deep-level characterization of protein expression and PTM. And I'm highlighting one of these such workflow using isobaric labeling. So by doing this, so you can have higher throughput and you can have really deep coverage so far. And we have been using this for many of our milestone works for several tumor types, and not only in our lab but also across CPTAC and other centers as well. But one of these obvious not advantage, but disadvantage in a way is that you still require lots of material to work with in order to achieve the comprehensive coverage. And in a case of PTM analysis, typically requires millions of cells per sample still. And so one of the issues with this is that such analysis only provides a population average and obscures the important cellular heterogeneity. So I think needless to say in the audience here, that it's really important to characterize these tumors at really fine detail level as possible to really understand the heterogeneity issue, which is of particular importance here. And this can also affect directly the patient's response to any treatment or therapy. So there's a lot of things we could do, right, using mass spec. But one of the obvious questions of using mass spec based approach for single cell analysis is it sensitive enough to do so? Actually, in theory, it is. If you run the calculation, you're supposed to be able to see 25 to 50% of the proteome in any given mammalian cell based on the current mass spec sensitivity. But there are a lot of sample processing and manipulation that's needed along the way in a typical proteomic analysis, because we need to extract the proteins from any specimen, and we need to convert it into peptides in a lot of cases for the effective analysis. And then there's a lot of contact in the process, which could lead to a huge amount of sample loss. So there is a lot of progress recently, just in the past few years, in terms of improving the analysis, of single cells or near single cells level detection using mass spectrometry, and touching different areas in along the way in analysis, including sample preparation, separations that's needed up front, and also sensitive mass spec and some other alternative detection methods, and as well as data analysis strategies. So I won't be able to give you cover all these different areas, but today I focus on the first part, sample preparation strategies and handling. So you can see from here, this review here, I just wanted to highlight two reviews, very nice reviews, very recent reviews as well, to cover this area. And one of our contribution is to miniaturize, the sample processing platform, which I'm going to talk about in the next couple of slides, and then some strategy based on isobaric labeling in terms of quote unquote amplifying the signal for protein analysis. So this is our strategy, basically miniaturizing the reaction space for this. The typical volume for this chip, each individual well is only 200 nanoliter, and we're using this setup. We can dispense and also withdraw the liquid automatically and completely from each individual well. And by doing so, we can minimize a lot of these surface contact cost, sample loss. And also we can automate a lot of these typical proteomic sample manipulation in the same device without the need to transfer to another vial or whatever. So to, again, minimize the sample loss. So we have been using this for a lot of upfront, technology in terms of transferring the single cells into the reaction vessel, the nanopots. In this case, obviously you can couple it to FACS sorting, very straightforward. You just need to adjust the nozzle space and flow rate in order to dispense a certain amount of cells into a given nano well. And also we can couple it to laser micro dissection. In this case, we utilize the DMSO and gives us a higher surface tension. And you can deposit this DMSO droplets in the nanopod chip and reverse it and use it to capture the laser shots. This is one of these earlier application we had using this nanopods workflow. So in this case, I'm showing you an analysis of single cells or a small amount of cells, in this case 20 cells, prepared from human lung tissue freshly. So if you look at the single cell analysis from each cell, in this case, we're using the label-free methods, mass spec methods to analyze the protein content in these cells. Like I mentioned before, all these sample handling from initial lysis to alkylation, digestion, and then final analysis. These are all conducted automatically within the same nanowell with a volume smaller than 200 nanoliter. So in the end, we were able to identify about 500 proteins from each single cell. And in the case of the 20 cells samples, each case we have about 900 protein IDs, and then the other things that we can capture the essence of the difference in these different cells. So the next step for us is to further increase the sensitivity of the detection. So one of the ways to do that is to sort of amplify, the signal from the sample using a so-called boosting strategy. So this strategy, I think the concept of this it started many years ago, back in 2010, and then later people started to adapt this for similar analysis. And then also, I think year 2018, we start to, not us, but another group start to adapt this technology for single cell analysis. And for us, we also look at this, but for different application initially. In this case, the strategy we call this boosting to amplify signal with isobaric labeling, or basal, was it to use it for PTM analysis, in this case for phosphorylation analysis. So you do the labeling individually to all your small samples, and then you label a larger amount of samples as boosting using any channel, and then any TMT channel in this case, and then you combine them so that in the eye of the mass spec it becomes one sample. And when the time you pick this precursor ion for fragmentation, it's actually the same species. That's the principle of boosting, right? But the trick is that once you fragment the parent ion, and then in the what we call reporter ion region, you do have the ability to tell which signal is coming from which specific channel, or in this case, which specific small size sample. You can do quantitation for the different samples, and in this case, you just ignore the signal from the boosting sample. So you kind of achieve both quantification and identification of these small cells in the same process. So we combined the nanopod and then the basal concept in this case, and we put this into a integrated workflow. So you basically do everything on the chip, including, like I said, previous lysis, digestion, and then also TMT labeling, and sample pooling into all one automated workflow. As you can see, we have a larger well here for combining all these individual samples into one combined TMT labeled samples for mass spec analysis. And as an initial application, we analyzed 72 cells from 3 different cell lines. And we also did bulk analysis as a comparison. As you can see here, we have reproducible, robust quantitation achieved from each individual of these individual single cells. And then, we can also largely recapitulate, the differences among the the bulk analysis at the single cell level, even though with higher amount of variation, which is expected. And in this case, we only using 25-fold boosting. And also we started to test, obviously as you can imagine, the higher amount of boosting you put in there, in principle, you should have higher coverage or higher increase in detection sensitivity as well. But we also start to see, at least initially, the quantitation quality start to deteriorate. So we ask ourselves why, and then how can we further improve this? So the limitation, obviously, is limited to by the total amount of ion that's feasible for the mass spec to accumulate and then transport to the next stage of detection. So in this case, in the typical thermal instrument, in this case the Lumos instrument, you're limited in the C-trap, accumulation capacity. So when you have a fixed total ion capacity, you have a fixed amount of sample ions that's allowed, and at any given moment. That's limitation. And then in the simulation type of experiment we did with this, it's just we did 3 replicas using 3 channels and leave all the others empty. And then we try to use a different amount of boosting material, in this case 10 to 200-fold increase, and try to see how does it change, right? And then as expected, the peptide level detection increase as you increase the boosting material. But the reproducibility in here, represented as the Pearson correlation across the 3 different replicas, is decreasing. And you can see the CVs as you go higher, the more deviation you have, from one replica to another. And again, that's it's pretty obvious that's by the limitation in the number of actual ions from the sample channels that's here as you go higher. As you can see that the 200-fold start to have appears to have the lowest amount of ions from the sample channels. So one way to do this is to increase the total ions that's allowed in the mass spec. So by increasing this, what we call the automatic gain control in the thermal instrument. By doing so, because once you increase the absolute amount of ions into the C-trap, you're also increasing the absolute amount of ions from the sample channel, even though the overall ratio from sample to boosting doesn't change, but the absolute amount of sample ions increasing as evidenced in this plot. And the effect it's pretty obvious. You can see as we increase the AGC from 5E4 to 5E6, the reproducibility improves and the variation reduced, and also the ion ingestion time as a kind of an indication of how much time you actually spend on accumulating, a certain peptide species, also from increase from a very low level to a higher level once you increase the AGC settings. But one of the drawbacks is obviously as you spend more time accumulating more ions, for a certain ion population, you basically reduce the duty cycle in the mass spec analysis. As a result, a decrease in the number of peptides that was peptide identification that we were able to achieve in the end. So there's a compromise, and then you need to basically carefully evaluate the optimal compromise a turning point in terms of identification and quantification. And so in this paper we published, we run a series of different conditions. We compare, even though it's only an initial set of comparison and systematic kind of evaluation of different settings, but that kind of give us initial set of optimized values in terms of boosting ratio, AGC setting, and then also ion ingestion time. So we call this improved basal approach. And if you compare this improved basal approach to the original basal approach, which is typical bulk analysis mass spec conditions, you can see there's huge increase in terms of identifications, and also the sample signal, from these sample channels, and then just this very simplistic view of the quantitation quality. You can see this iBasal is improving the quantitation quality quite a bit, judging by the reproducibility across this replicates of the 3 different AML cell lines. And just a very recent paper, I think people start to realize the importance of this type of work, and then start to look at more fine details in exactly how you can fine tune your analysis. So this is just one of our initial application of this workflow to the detailed analysis of single cells from 3 different AML cell lines. And you can see in each one of these AMT analysis, we can identify about pretty close to 2,000 proteins. And across a total of 13 different sets of TMT, we have in terms of quantitation. And if you call these 70% or more without missing value, we were able to identify over 15,000 quantify 15,000 proteins, and then achieve good reproducibility. And then the single cell data actually were able to reveal the major differences of the 3 different AML cell lines as expected. This is pretty similar to what we observed at the bulk level in terms of the depth of coverage needed to review these differences. Another application that we've done is to apply this to characterize different plasma cells from a multiple myeloma patient. So here I'm showing you the single cell RNA-seq data. Obviously, you can review a lot of these differences amongst a lot of different cell types that you can get from a typical bone marrow sample. But when you look into details of just the plasma cells through their mutation mapping, you can also classify them into several different clusters. So we wanted to be able to achieve the same type of characterization of the single cells at protein level. So we basically applied the same iBasal workflow. Here I'm showing here's the first stage. We try to isolate the plasma cells using CD138. So that's the plasma cell population, and that's everything else. And that's a result you can get from a typical bulk analysis. Not a huge amount. We start from 5 nanogram using a typical basal, kind of a condition. You can tell the difference. But once you start to use this look at the single cells and of the plasma cells, and then also in this case another isolated T cell population, you can see we can cluster these cell types very clearly. And if you look at the profile, and I wanted to highlight these, in this case, the T cells. There's 2 cells that's far away from all the rest of it. You can see we can capture these differences very nicely on the protein profile in these 2 cells. And same as in the in plasma cells, there's definitely differences across different plasma cells that we will isolate for using the FACS sorting. So one of the questions is, can you use even higher boosting and achieve better results? So the answer is yes, but how much higher you can go, right? So here we test the extreme condition of a 1,000-fold. And as you can tell, we can still, get pretty reasonable quantification by applying this iBasal strategy. Obviously, they're slightly worse than the lower boosting level. The gain that you get from 100 to 1,000 is not much, but there's still a slight level increase. But one of the things you sacrifice is the dynamic range. It's definitely more compressed once you go higher. And also, there's other people recognize the importance of this issue, and there's more systematic, characterization of such issue in the recent Nature Methods paper. I recommend you to go over there for more details. And here, I just wanted to show there's also another way to improve the overall detection, which is fractionation, using nanoscale fractionation. We have this nice system developed, we call this nano-level fractionation or nano-FAC. You compare it, we were able to increase the protein identification further to over 3,000 and still be able to quantify 2,000 of them, and then still maintain, actually increase the at least same level of quantification. And another thing, I realized the time is getting close. There's other things I wanted to cover, so I wanted to go quick. But just to quickly point out, the TMT Pro obviously can increase the multiplexing, and we know it's very similar to TMT. I just wanted to point out, you can actually analyze the data together, meaning you can, if you want, you can analyze the single cells with TMT and TMT Pro and still be able to analyze the data effectively in terms of quantification. So in a way, you can increase the overall scale of your work by using these different labeling strategies. And again, I don't want to look into details in these, but there's two very nice new research articles just recently deposited into bioRxiv using alternative single cell workflows. In this case, there's a EvoSAP based workflow. And then here there's a FENS Pro based workflow. In each case, they were able to identify and quantify about 1,000 proteins per cell. I recommend you look in details over there. Just wanted to quickly point you towards some of these what I call new single cell phosphoproteomics work that we've done. Excuse me. So here, just kind of a quick thinking, right? Some people say, why don't just go straight ahead, boosting to 1,000,000-fold, try to see whether we can analyze the phosphor content, in a single cell, right? That's what I'm showing you here. The simple answer is you can't, because you can't get enough identifications from such setting, even with the iBasal approach. But you do, you can use 1,000-fold boosting and then get down to 200 cells level for cell typing. I think it's still feasible. In this case, you have only roughly 700 sites identified, but you can still cell typing. You can still do cell typing experiments. So one way to further improve this, obviously, is to improve the upfront sample processing, right, in terms of sensitivity. So what we did here is what we call this lab in tip. So basically, you try to do everything in tandem in tips, and in doing so, you minimizing sample transfer to increase overall sensitivity. And so what we can see here, get down to 1 microgram level of input protein, we can still identify about 2,000 phosphopeptides using just MS-based identification. So here, we also try to increase the overall AGC 1E6, because this is a phosphor analysis. The complexity of the sample is not as limited as the global sample. That's why not only here we increased AGC, but also increased ion injection time from 300 to 500 and then 1,500. Try to compare the results. And as you can tell, 1,500 milliseconds definitely give us better results here. And then in this case, if we fix the overall input to 1 microgram, we can do 1,000-fold boosting and get down to analysis of 10 cells and still achieve reliable quantification about 100 sites, or over 1,000 sites in 100 cells. And in terms of the quantification, is still pretty reasonable. So as you can see, we still capture the differences amongst the different cell lines, and also maintain this robustness in terms of reproducibility within the same replicates of the same cell lines. And if you pick the overlapped phosphopeptides in this representation, you can see there's basically it tells you the sites that we capture from the 10 cells are still reflecting the differences that we capture at the 100 cell level, which is shown here. And obviously, we can still try to do for the nanoscale fractionation. In this case, there's 2 ways to do it. You can either use the NanoFact automated system or using the manual level in tip fractionation. Here, I'm just wanting to show you a high level kind of in general, this is a quick experiment, pretty new. We compare actually the manual mode to the automatic mode. at least for now, we're getting better results from the manual mode, higher ID and lower overlap in terms of fraction to fraction overlap. I'd say this is probably still the automatic system can be further improved by fine tuning some other components. I don't want to go in detail here, but if you project this level, if you start with 25 microgram of material and then you do 1,000-fold boosting, you can get down to 200 excuse me, 250 cells analysis and achieve quantification of 15,000 phosphopeptides. So just to kind of outlook, that's kind of my final slide. We wanted to, again, apply this lab in tip concept, and also the possibility of combine this process with the initial nanopods processing, at least for initial digestion, and then you can deposit the samples in the tip and do IMAG everything in the lab in tip setting, and also using the plate platform and try to automate this system with some other commercially available systems. That's kind of a work in progress. So I don't think I need to go through the summary again, but I guess the message here, obviously, there's a lot of advances being made in the space of single-cell proteomics or neo-single-cell phosphorylation analysis. And for me, I'm very optimistic that we will be able to apply a lot of these in larger scale applications already. Some other people already start to apply this, try to address some of the heterogeneity issue in certain systems. And I'd be just stopping here and acknowledging two people in my lab who made really tremendous contribution to the work I presented here. Chai-Feng Chai and then Yin Zhu, and then many other people like Ma Wei-Do, who's now at Thermal, and then Ryan Kelly, who's at BYU, started this work earlier. And our collaborators for the multiple myeloma work, and then funding from NCI, including CPTAG, EDRN, and some other like BTR and the LONMAP program at NIH. Thank you for your attention.
Thank you, Tao. That was a great presentation. We are a little over time, but I think I'll squeeze in just one question and we will conclude. Let's go with this one. Okay. Which tool is suitable for single-cell proteomics analysis? This particular person uses Seurat for single-cell RNA-seq. What would you recommend?
Could you repeat? I didn't get the first part. Is which platform?
What tool is suitable for single-cell proteomics analysis? So they for single-cell RNA-seq analysis, they use Seurat, S-E-U-R-A-T.
Mm-hmm.
So
right. Again, I'd point. So I guess you are asking which commercially available tool, right? Our platform, the nanopod system, currently is still not commercially available. But there are some other people, nice work reported by, I think I go to this slide. There's two papers recently deposited into bioRxiv. I highly recommend you look at those. One of the system is based on the EvoSAP system, and then also the front end 384, the PCR, plate, if you will, for the front end sample handling. And then there's another work using very similar setup, but they're obviously using slightly different, conditions in terms of sample lysis, and then initial processing. But both of these, this work is a label-free workflow, and this one is a TMT workflow. They were both able to achieve, 1,000 protein IDs. So I'd highly recommend you look in details in these two papers, in terms of commercially available systems for your analysis.
Okay, and then I'm just going to squeeze one in really quickly, one more. Can you use this system to do immunoprecipitations and study protein-protein interactions on a single-cell basis? Sounds pretty ambitious.
Yeah, I wouldn't say it's impossible. We can try. Certainly, but again, the key is upfront. Sample manipulation, as alluded on, at first couple of slides. In terms of the mass spec sensitivity, you can achieve very high sensitivity detection, as long as your proteins of target don't get lost during the sample process. There's a chance. And the other way to do this also is to run this in a targeted mode. So we are also doing some of these targeted single cell work. We actually demonstrate for some of the targets, for example, targets in the EGFR pathway, we can analyze them at a very high level of sensitivity for including some of these phosphate events.
Okay, wonderful. Well, thank you so much. It was a wonderful talk. I'm going to take back the ball, and we're going to conclude our first presentation for the year. Dr. Liu, thank you again so much for a great talk. And thank you everyone for joining us. We have another webinar coming up in the middle of February with Dr. Sam Payne, and we hope that you can join us there for that one as well. And with that, I'd say thank you.
Thank you, everyone. Bye-bye.
Okay, bye. I'm going to share my screen.